Good afternoon and welcome to the second showcase of African Soil Information Community of Practice. We are really thrilled to have each and every one of you today with us, and we are delighted to share this time together with you. Today is about the showcase for the latest works that have been done in Africa on the lab analysis domain in soil information workflow. We hope today's program will be informative and inspiring you. Today's event is about coming together, and even if it's virtual, to connect, learn from each other, and perhaps to make new collaborations along the way. So the cooperation with the national level soil information system is consists of community of practice and a resource library that we've been working on in ISRIC. The community of practice is a community a platform that provides professionals and researchers in soil and soil experts a platform to exchange their idea, insights, and discuss challenges and contribute their understanding of soil data and information across Africa. As you can see from this map of Africa, the green areas are the countries where we have members and ambassadors, and we also hope to reach other countries so we can fully cover the whole continent. The goal of this community is knowledge sharing, foster collaboration, and also education via online resource library and capacity building training. And we also hope that people can connect, building bridge between all the experts across Africa and improve the network for all soil scientists. At the moment, we have 624 members from 31 countries, and all new members from all African countries are welcome to join us. Either you can visit our website that's you can see here, or you can scan this QR code and then you can fill the registration form. Then you will be added to our platform. The platform at the moment is a WhatsApp group. And then hopefully we can move to a better online platform. The activities we do are webinars, question and answer sessions, joint publication, ask the expert sessions, and we also do spotlights of our members and ambassadors. We arrange capacity building trainings, and we do showcases per each domain every month. We have 29 community of practice ambassadors or community leaders from 6 countries, 6 African countries. You can see the profile of our community ambassadors through this link, which is also in our website on isric.org under Community of Practice. Other online platform that we have is the website, as we mentioned, in isric.org and the Community of Practice. You can also visit Community of Practice on LinkedIn on this website. And as I said, if you can fill the online form, registration form with a correct phone number, then we can add you to a WhatsApp group. I am the community manager, and you can also visit my profile on isric.org people for more information about me and my publication. So I will stop sharing now, and we can start with our first speaker today. We have Dr. Nandomiso. She's a soil scientist working for Agriculture Research Council, Natural Resources and Engineering in South Africa. So the floor is yours, Dr. Nandomiso.
So my presentation today is going to take you through the wet chemistry and spectroscopy aspects that are involved in the Soils for Africa project. We'll be using the Soils for Africa project as a case study. There's many projects that we do in our lab, but for this specific presentation, we chose Soils for Africa. But I am aware that some of you may not be familiar with the Soils for Africa project, so I put this slide to give you like a bit of a background. There is more information if you go to the link on the slide when you get some time. But this project aims to address the need of fragmented soil information that exists in Africa. Most of the data that we have available was collected between the 1960s and the 1980s, and recently in the ISRIC project. But all of this data is difficult to harmonize because of different methods that we use. So this project was trying to iron that challenge out with the aim of developing an open soil information access that will have reputable soil survey and soil analysis methodology and information. These are all the partners that are involved in the project. And then, as you can imagine, the project of this magnitude involves a number of work packages. So for this project, we have about 7 work packages as listed in the slide. Some involved in the coordination, communication, and the outreach of the project, stakeholder engagement. And my specific work package focuses on soil analysis and capacity development. So for this work package, we are collaborating with World Agroforestry Research, shortly known as ICRAF, based in Kenya. We also collaborate with IBEC, based in Greece, as well as ISRIC, based in the Netherlands. The specific aim that our work package addresses is to carry out all the required analysis on the soil samples that will be collected during the field campaign to acceptable levels and quality and reliability of these samples that will be collected in the project. So for those of you that may not be aware, the project intends to collect about 20,000 soil samples across the African continent and at least 10,000 soil samples from the lower depths, depending on the pedogenic area of the soil. So the specific objectives that we address in this work package is to prepare the laboratory facilities specifically for the spectroscopy, because this is the area that our laboratory was not familiar with before. The Agricultural Research Council has the reputable wet chemistry laboratory that has been running for more than 20 years and uses ISO certified methods. But some of the analysis that was required by the project were not part of our routine soil measurements. So then there was a need for training for all those areas, as well as preparing the SOPs for all the analysis that will be done. Like I mentioned before, the aim of the project is to share this information with the Africa continent at large, and then to install and apply quality control procedures, and eventually produce wet chemistry and spectral soil analysis results, calibrate the spectral results and interpret them. So to address the first objective, which was to prepare the laboratory facilities specifically for spectroscopy, there was a need to identify a laboratory that will be fitted with a functional air conditioner, and to procure a functional spectrometer, make sure there's space to archive these samples after the analysis has been completed, procure the milling instruments to mill these soil samples to a specific fineness. As I go through the presentation, you will realize that the preparation of wet chemistry and spectral analysis is not entirely the same. And also to train the staff on how to use all these equipments that would have been procured. The second objective was to train the laboratory staff on spectroscopy. So we received our training from World Agroforestry Research based in Kenya. They physically visited the Agricultural Research Council in 2022, following a series of online meetings trying to get us up to speed with the concept of spectroscopy and spectral analysis as a whole, and conducting the actual measurements. Then we were also trained from the basics of receiving the samples, packaging the samples so that they remain traceable in the lab chain, and preparing them for analysis, understanding the good spectra and the bad spectra so that we produce good quality results. So I just put some of the pictures to show you some of the training that we went through. Then the third objective was to prepare the standard operating procedures. So like I mentioned, the spectral analysis was not part of the Agricultural Research Council laboratory before. So we adapted our SOP from World Agroforestry Research SOP to adapt it to all the instruments that we have in our lab. And when it comes to wet chemistry analysis, there was already a lot of ISO certified methods for some of the analysis that was requested by the projects, but some of the analysis which were not ISO certified, the SOPs were developed from national books that are currently used in soil science, as well as well-cited scientific papers, as you can see in the slide. For the fourth objective, which is to ensure quality control measures. So I'm going to start with the wet chemistry aspect. The quality control measures are involved from the beginning of the process where we receive the samples. So for this project, we receive samples in different shapes. They come either in boxes or different packaging, if I should say. And the example in this picture was the batch of samples that came from Nigeria, and they are required to be double bagged so that they don't leak, or if there's any leakage, there is no cross-contamination by all means. And the country supervisors share the list with us before the sample arrived to our lab. And then we also have the database, which is called survey and data management database, where we can download the information that was loaded in the field when they were collecting the samples. And we use that information to verify the samples when they get into the lab. So when the samples get into the lab, they are transferred into these plastic vial containers, and we put the QR code that come with the sample as a way of keeping it safe for future use and for future reference. Then they are registered into the ARC's South Africa lab numbers in our database. Then some of the analysis require that the samples are finer, for example, nitrogen and total carbon. So then we put this small plastic bags, if you can see in the slide, it's just to sieve and include that soil that is going to be used for that specific analysis. Otherwise, most of the samples come already dry, prepared for the routine analysis, which requires just less than 2 millimeter sieving. And then when it comes to the internal quality control for the actual laboratory, we make sure that we are working with well-serviced instruments, calibrated balances. Some of these instruments require annual or biannual maintenance. And so we keep up to date with our calibration schedules through our ISO certified suppliers, as well as in-house verification. Then we use the chemical reagents that are NIST traceable and also purchased from ISO certified producers. And we make sure we keep all these chemical reagents according to the producer's guidelines. Like I mentioned before, the methods that we use are ISO certified. They are also relatable to the Lucas procedures, if you are familiar with the Lucas procedures. And then make sure that the people that conduct this wet chemistry analysis are competent. Okay, so when it comes to the quality control that are involved in soil analysis, So we have an area where the weighing is done, an area where the extractions are done, so that there is never a confusion of what's going on at a certain time. Then we have a temporary storage where the samples during the analysis in the lab, they are stored. So each technician knows that for as long as these samples are not out of the lab, they can be found in this area. And we have logbooks, SOPs, technicians at all times during the analysis. So whenever there's a problem, the technician is able to page through the SOP and figure out what could have went wrong, just to make sure that there's traceability at all times. Then when we get the results, we still have our internal quality control procedures. So sometimes we get the results in Excel formats, generated through the instruments, or sometimes they're just in hard copies. For example, when we do pH, EC, things that are not computer-based. And then the chief technician responsible for the entire wet chemistry projects checks for outliers, checks for the repeatability of results, and generates the reports based on the client's preferences. So in this case, for the Soils for Africa projects, we were requested to provide the reports in a CSV format. So our final report is always in a CSV format as requested. Moving on to spectral laboratory, we also have internal quality control procedures from the beginning of the process. When we receive the samples, like I mentioned, they come in different packaging, as you can see. And then we have the list that the country supervisor would have shared with us. Then we have this survey data management database where we download the information from the field. Then we conduct sample sorting just to make sure the IDs that are written on the list that we receive with the samples matches with what is in the database. And then we identify discrepancies if there are any, and we come up with the updated list, which we will share with the country supervisors and ask for clarity if there's a need. After all that is done, then the samples are ready for milling. So what we usually do for the spectral laboratory, other than using the miller that I showed you earlier, we have these agate Heslin mortars. They are very easy to work with. They are easily cleaned, so there is no issue of cross-contamination. And then we put them in clearly labeled containers, which will be used during the analysis. And then we package them in these alpha cups. They are very small, specifically used for this spectrometer. And we make sure that the alpha cup that we have filled is next to the container that we'll be analyzing, and the container is well labeled. It's clear for the technician that will be working in there to type it out or scan it. So moving on to the actual analysis. There's a list of steps that are involved according to the standard operating procedures, but just a few highlights. There is a need for the laboratory to be set at 24 degrees Celsius. That's why we needed to have that air conditioner functional. And there's a need to have the laboratory clean at all times because the instrument that we work with is very sensitive to dust. And then there's also a need to use the gold standards that come with the instrument, to use the quality control samples that we have in the lab, like the samples that we already know the wet chemistry results, as well as we have 3 soils that we collected from World Agroforestry Research that we used as our calibrating standards during the analysis. And then, so we capture the ID, then we conduct our analysis in OPAS lab. So if you are familiar with the spectral or spectrometer, you would be aware that it can analyze any type of any material, if I should put it like that. It can analyze water, it can analyze plants, But for this specific analysis, we use the drift module, which is optimal for our soil analysis. And then we visualize our spectra each time after each analysis to make sure that there are no carbon dioxide or moisture absorption peaks that we should be aware of. And at all times, our moisture or our humidity in the instrument should be reading at less than 20%, just to make sure that we get accurate results. So we keep it fully desiccated. So we put a clean desiccant at all times once we realize that the one that's in the instrument is now almost at 20% moisture content. And then after that, I work together with the interns to check the IDs if everything has been analyzed properly before we move on to the next page. So we transfer our files from Opus files, and then we use RStudio to process our data. We have multiple scripts that we got from ICRAF as well to process the data, and then we share our results in a form of a flat table in a CSV format, the same way as the wet chemistry. Then we have also other requirements from ISRIC, our partner, which is responsible for the soil information system, that they want us to keep the identity of the samples, the source for Africa format. You can recognize it from the screen with the country code, the PSU number, TSU number, and the secondary unit and the tertiary unit, as well as the depth. And then each batch that we receive, we rename it accordingly so that we avoid mixing up. This is just, because of time, I'm gonna really go through this quickly. This is preliminary soil results that we have generated so far. And from these slides, you can see that you can tell when there are discrepancies, you can tell if there's any soil sample that was not analyzed properly. And then we did a statistical analysis. This is a T-Hotelings diagram. Usually it's used to identify outliers, but in this case, we just want to see if there are any clusters in our data. Should we be worried about these samples? But after we rerun the samples and we realize there's nothing wrong with them, then it just tells us that they are clusters. So as we collect more results, or maybe at the end of the project, we want to see if this is going to change or how is it going to look. And then we are able to clean up our data using those scripts that I told you about. We are able to check the precision of the spectra, because remember, the aim is to make sure that we have repeatable results. And then we are able to overlay our wet chemistry results with the spectral results to see that our field campaign design method as well as our analysis method is in line, and it can represent our data set. So these are some of the models that we generated. You'll see most of them are fairly poor because we are still at the beginning stage of the projects, and we'll keep retraining these models to improve them over time. This is an example for clay. You see the first diagram has higher clay contents, but if you remove them, you see that your model improves and you see the statistics quite improved. But this is something that we'll be doing at a later stage. I just put here in the presentation to show you what we are capable of doing with the data and that we can generate reliable models. So going forward, we will be collecting more spectral data as well as with chemistry data, shipping some of the samples to our collaborators in SGS and ICRAF in Kenya for them to run the analysis and tell us if we are still getting the accurate results and hopefully generate more improved, robust models that I can show you in future. Thank you.
Thank you very much, Dr. Nandamiso, for the interesting presentation. I would remind our guests to kindly post all your questions in the chat, and we will address it after the all presentations. So thank you very much once again. And we shall move to our second speaker, Dr. Albert Kobina Mensa. He's a soil science researcher from Council for Scientific and Industrial Research, Soil Research Institute, Ghana. Dr. Flores is yours.
Thank you so much for the time, and it's my pleasure joining you all the way from Kumasi in Ghana. My name is Dr. Albert Kobina Mensa, and I'm speaking from Ghana. I work with the Soil Research Institute here in Ghana as part of the Council for Scientific and Industrial Research. I did my PhD in Bochum at the Royal University, Bochum in Germany, and I did my master's in Kenyatta University in Nairobi, and I did my bachelor's in the University of Cape Coast in Ghana. I am privileged to present to you some of the works that I've done and how to use a thorough strategies to be able to talk about how redox chemistry alters mobilization of arsenic in a contaminated gold mining site. So we have mining. First, the open pit is dug. Then after the open pit, the ore is extracted from the pit and is taken via transport to the mine processing site. Then from the mine processing site, at the end of the day, the effluent or the waste is stockpiled. Then as the waste stockpiles, we have this stockpile being carried by the sand or being carried by wind into water bodies and also into the atmosphere. So now these are some of the sources of mine waste, or sources of what is called the potentially toxic elements that are generated from mining sites. So we have this particular one, so that is mine waste stockpiled at a mine site in Ghana. Then we have this one, as we can see, poorly stored, and when it rains, all these things are going to carry heavy metals into the environment or into the food chain, and also into available groundwater and surface water. And it can also be inhaled when it becomes airborne. And also children can also come into contact with some of this, and they will experience deleterious health consequences. So this is also, as we can see, poor disposal of mine waste from mining site in Ghana. I took this when I was doing my PhD in 2020 at one of the mining sites here in southwestern Ghana. So as we can see, all these heavy metals are impacted by redox chemistry, and when the land is left abandoned without reclamation, it becomes a playground for children. And like what we can see over there, this particular site is a site that contains arsenic of almost 8 grams per kilogram. And we find children playing on such contaminated site, and they are exposed to all the heinous consequences as far as toxic element, especially toxic element are concerned or is concerned. So then we have, so there was this paper that talks about mercury content in river sediment, 3.36 milligram per kilogram, which is actually above the stipulated threshold, which is somewhere around 0.05 milligram per kilogram. Arsenic content of 183 milligram per kilogram, and I have found levels of almost 9 grams per kilogram. And so this is like a disaster waiting to happen, you know. So like I had already stipulated, if this result in food chain contamination, we love our rice. For instance, we eat a lot of rice here in Ghana. And mind you that rice is mainly associated with contamination with arsenic and cadmium. more so because rice is grown in a wetland or in a paddy field. And this paddy field experiences a lot of redox chemistry, and all these tend to contribute to mobilization of toxic elements into our rice. So when you Google rice and arsenic, you realize that rice is associated with arsenic pollution a lot. The reason being that of what we are going to talk about today. So contaminated site is really a very big problem. Then when you go to Bangladesh, we find that the people they are mostly exposed to arsenic poisoning that we can see in A and B. And this arsenic poisoning, especially in Bangladesh, is associated with their groundwater, arsenic being actually contaminated in their groundwater. Now, what is the redox effect on this heavy metals or on this potential toxic element? We could see that mobilization of arsenic could be altered under different redox chemistry. We can see that, for instance, wetlands, watershed, floodplains, flooded and waterlogged ecosystems, paddy fields, high rainfall tropical ecosystems are mostly associated with redox chemistry, and that all these will have deleterious consequences in actually causing the mobilization of arsenic and other particularly toxic elements of concern. So when we're able to study the dynamics of arsenic under redox conditions, it could help us to really come out with the appropriate remediation measures for arsenic contaminated fields. For instance, arsenic 3 is more toxic than arsenic 5. And arsenic 3 is more of a reducing forms in wetland chemistry or on wetlands, and also in flooded ecosystems. You find more of arsenic 3 than arsenic 5, because in wetland ecosystems, arsenic 3 is more reduced form of arsenic 5. So in our lab in chemistry, where I did my in Wuppertal, where I did my PhD, we have this particular, which is called a modified microcosm, a modified automated biogeochemical microcosm setup. And this setup over here is used to study the redox chemistry and how redox chemistry could actually affect mobilization of toxic elements into the available food chain and groundwater. So I will not spend much time, but I will use the pen. I'll use the pen here. So first, it starts from here. This actually, this is a setup. It's like a jar. I'll show you. This jar has a stirrer. So there's a stirrer here. Then, so in this jar, you put in soil of somewhere around 200 milligrams, 200 grams. Then you put in water times 2, or 1:8. You put in water of somewhere around 1,600 liters into this. Then what you do is that you put in a stirrer and you on the machine, and the stirrer stirs it continuously for like a month. Then you put in probes, some measure temperature, some measure pH, some measure the redox potential, which is the EH. Then everything is recorded in what is called it's recorded by this machine. Everything is recorded by this by the laptop or the computer. Then we also have a system to increase the reduction. So to increase the reduction, so we have nitrogen being pumped to cause reducing conditions, and we have oxygen being pumped to create oxidizing conditions. So that is how it works over here. Now, so like I have demonstrated, we have the microcosm setup there. The jar is filled with soil, and we add water to it. Then at the end of the day, so we have water being added to the soil to make slurry. Then at the end of the day, we add glucose to stimulate the activities of living microbes so that they can cause the redox reactions to occur. Then you put in, like I said, 200 grams of air dry soil. Then you put in 1,600 milliliters of tap water. You put in glucose. Then you put in, so then there's constant stirring. You flush with nitrogen and oxygen to control the EH values. Then there's automatic recording of EH and pH. Then at the end of the day, you take this syringe, you then to withdraw, take the syringe to withdraw, some soil slurry. Then this particular one is 90 milliliters of soil slurry using the syringe. Then you do infiltration, or sorry, you filtrate and do everything in a glove box. So you do everything in the glove box and using a liter paper of 0.45 micrometers. Then everything, then the extractant are ready for analysis. So you also, then you analyze for arsenic, you analyze for iron, you analyze for manganese, sulfur, DOC, which is dissolved organic carbon, and other parameters that could affect the biogeochemistry of the arsenic or the element under study. So we did this experiment here. I did it in Bochum, I did it in Wuppertal with that particular system or setup. So over there, I quantified the effect of EH-pH changes on mobilization of arsenic in the contaminated gold mining spoil. So that is what I did. Now, this was the results that we are going to get. So you will see that we are going to have changes in the pH, and we are going to have changes in the EH. This point you see are various sampling points. There are various sampling points, the blue at various sampling points. So we take measurement at every 6 hours after incubation. So we can see that whenever from the beginning, the pH fell sharply, whilst the EH also fell sharply. The reason being that during this time, they were active, the microbes were active, so they were producing more of carbon, and the carbon resulted in formation of carbonic acid, and carbonic acid caused the pH to drop. But after some time, from the 40th hour, we could see that it started rising steadily. Then the pH also started. So wherever you see pH rise, EH will also go down. So it's some sort of negative relationships. I will explain this as the questions come up. So that's what we can see. So I've explained all these things. So there was an inverse relationship between EH and pH. pH sharply decreased after flooding from around 340 millivolts until negative 50 millivolts, ATF incubation hours. Then we can use this to study the redox potential on this, particularly the arsenic mobilization. So this is arsenic here. Then we can see all these parameters: arsenic, ion 2, ion 3, manganese, aluminum, sulfur, sulfate, chloride, DOC, silver, pH, and EH. All of them bear influence on the mobilization of the arsenic, which is this one. So to make long statement short, we can see that wherever arsenic or wherever EH goes up, arsenic mobilization drops. Wherever EH comes down, arsenic mobilization increases. So we can say that arsenic mobilization increases with decreasing EH. The reason being that when we have decreasing EH, we have more, or what is called, proton is consumed. So when proton is consumed, you have more hydrolysis, and the hydrolysis causes the ion or the arsenic to become mobilized. Then also under reduced redox conditions, we can see that the arsenic, the ion, which the arsenic is bound to, the ion reduces to ion 2. So as the ion reduces to ion 2, from ion 3 to ion 2 under reducing conditions, the arsenic that are bound on the surfaces, or those arsenic that are co-precipitated on the surfaces, will also become mobilized. So we can see that dissolved arsenic was high during the reducing conditions than in oxidizing conditions. Then we have dissolved Fe increased under reducing conditions. So any arsenic that is bound on the surfaces of the ion will also become dissolved and become mobilized, because there was negative relationship between the Fe and the EH, just like the same relationship between the ion and also just like the same relationship between the arsenic and the EH. So there was a negative relationship also between pH and Fe. So the flooding resulted in reductive dissolution of ion 3 into ion 2. So that caused the release of the arsenic that are bound on the surfaces of the ion. Then we also use what is called a synchrotron radiation science to study the speciation of this particular arsenic. So we took samples from box sample. We sampled at negative redox potential. We sampled also at positive redox potential. Then we sampled at the middle at 0. So at 0, we sampled. Then we wanted to see the species that are available at 0 redox potential, at negative redox potential, and at the high redox potential. We did this one in Taiwan at the beamline TLS7A. So then after that, we did linear combination fitting using using what is called Athena. Using what's called Athena. The major species that were found were sclerodite and arsenopyrite. So we used it to do the linear combination fitting. So this is the linear combination fitting results from the speciation analysis. So we can see here that whenever you see this color yellow is sclerodite. Then whenever you see green is ion arsenic humic or arsenic 3 ion humic. Then whenever you see pink is, we can see that when we started the experiment, the box soil contained higher percentages of what is called the arsenopyrite and the sclerodite. It contains higher percentages of arsenopyrite, the sclerodite, but more of sclerodite and arsenopyrite, more of sclerodite and arsenopyrite. Then when it was reduced to negative 220 millivolts, we can see that still the dominant species were more of sclerodite and arsenic 3 ion humic. Then at zero redox potential, we had the dominance of sclerodite and more of arsenic 3 ion humic. Then under oxidizing conditions at positive 220 millivolts, we have the dominance of sclerodite as well as the arsenic 3 ion humic.
Dr. Albert, sorry for interrupting you. Just to notify the time, please. You have 2 minutes.
Okay.
Thank you.
So we can say that flooding, leaching, runoff, rainfall, all will create and introduce reducing conditions in contaminated sites, especially gold mining sites that are abandoned, and they may increase the potential of arsenic release into water courses. We therefore recommend that various remediation measures should aim at the reducing mobilization and the release of arsenic from the site, from the mining site. So that means that we should really make sure that we don't cause wet conditions on the mining site, so that because wet conditions will sort of create conditions or create the enabling environment, which is going to cause the mobilization of arsenic. So we should target more of dry conditions than wet conditions. Thank you so much.
Thank you very much, Dr. Albert, for the informative presentation. And we have the final and last speaker today, Dr. Samuel. He's also soil scientist in and he's a technical coordinator of Soil for Africa field campaign at IITA from Nigeria. Dr. Samuel, the floor is yours.
Thank you very much. And for this opportunity, and I'm happy to be here. I'm Samuel Misili. I work with IITA. I'm a soil scientist, but I work here as a project coordinator, helping to support the implementation of the field campaign, which is for the Soils for Africa project, of which Nundu Miso has given a nice introduction in her presentation, so I will not dwell much on that. This afternoon, I have the pleasure of presenting this topic, which is implementing a continent-wide protocol for field survey and implications on soil data quality and integration. So in the next 15 minutes, I'll be making this presentation, and I hope you enjoy it. So briefly, we already have the introduction, but briefly, the Soils for Africa project, I'm using it as a case study, is a project that's about 4 to 5 years, and it has to do with developing soil information system for soil quality monitoring of agricultural land in Africa. It is a product of African Union and EU collaboration. It was funded as part of the Horizon 2020 program. We have 17 partners, as said by Nundu Miso in our presentation. And ISRIC is the proud coordinator of the program. IITA, my organization where I work, we are coordinating the field campaign in terms of the capacity building and sample collection for the whole of Africa. It is expected that the project will live till May 2025. And here on the right-hand side here, you see a map of our area of operation so far. The deep blue are the areas where we have started, and more or less, some of them we have completed the survey. And the light one are areas where we still have agreement to do the work, and we are planning to do, but soil sample collection have not started. Now, how can soil sampling methodology influence the analytical results? The methodology for soil sampling plays a crucial role in determining the analytical results obtained from laboratory analysis. And here is how the sampling methodology can influence the analytical result. One, in terms of the representativeness of samples. Proper soil sampling involves collecting representative samples that adequately reflect the variability of soil properties within the sampling area. Inadequate sampling, such as collecting samples from non-representative locations or depth, can lead to biased results. Then we have a sampling depth and profile. The depth and profile from which soil samples are collected can significantly affect the analytical results. Different soil horizons may have distinct characteristics, and sampling across the different depths allows for a more representative understanding of soil properties and nutrient distribution. Sampling equipment and technique is another method, is another factor that can influence the analytical results, and that is the choice of sampling equipment and techniques can impact the integrity of soil sample. For example, using clean and properly calibrated sampling tools prevent contamination and ensure accurate representation of soil matrix. The same thing has to do with soil homogenization. If the soil is not properly mixed during sample preparation, contamination can also occur and affecting the reliability of laboratory results. Sample preparation and handling are also key, and if not done properly, it could compromise the accuracy of analytical results. Soil sample processing and preparation are also important criteria to consider, as well as the laboratory analysis method itself. You hear when Demiso was presenting the analysis method used, she clearly indicated that those instruments were properly calibrated, and the officers that were to use them, they were properly trained. These things are very important to have a reliable result. Then the quality control measures too, implementing quality control measures throughout the sampling and the sampling process is essential to verify the accuracy and reliability of results. This includes using certified reference materials, conducting duplicate analysis, and regular calibration. Then, what is the structure, or how should the structure of this type of a protocol be that will go around the whole of Africa? So, the protocol one, the structure should be in form of a standardized sampling methods. So, by establishing standardized method for soil sampling, including the depth, location, and frequency, and utilizing advanced technologies such as GPS. We also use MapsMe in the South for Africa, remote sensing, and mobile applications for data collection. It helps to improve accuracy, efficiency, and consistency of soil data across diverse geographical regions as we have in Africa. This ensures that consistency across different regions align for easier comparison and integration of data. Then we have quality control measures. And these quality control measures in the South for Africa, we develop an online tool that we call SDMT, that is Survey Data Management Tool. And with this tool, you are able to manage and have a real-life monitoring of the activity going on in the different locations. So rigorous quality control measures to ensure accuracy and reliability of data is very important. And of course, it also involves training field technicians, using calibrated equipment, and conducting regular checks. Then the data collection parameter reserves, a number of data are being collected both in the field in terms of the soil surface characteristics, the slope, the topographic factors, the land use and land cover characteristics, the land management and water conservation practices in the field as well as so many of these properties are collected with clear specification on how they should be assessed. Then we have validation and calibration. We validate survey results through independent verification and calibration with laboratory analysis. And again, we also do this through the national representative, which we call them the country supervisors, who have to validate that, okay, these results collected are correct based on their experiences. Here is a sample of the electronic collection forms. Then the survey data and management tool I mentioned earlier. So it's an online system and has 4 or 5 components. The onboarding of surveyors, where people who are involved in the survey job, in the survey activity or the project are onboarded. The sampling points, where you can see all the sampling points and you can validate them, where you can assign job, monitoring, you can do monitoring and quality control as well as reporting. And this system uses a barcode system for verification of the soil samples, and this is passed on directly into the lab for analysis and reporting. The verification and quality checks are also done as part of the process. It has an inbuilt and as well as dashboard to monitor the status and progress of the activity that is ongoing. Now, in doing this, of course, we know for any important activity such as this, there will always be there's always be challenges. And here are some of the challenges. One, the diverse soil characteristics. Different regions within the continent, particularly in the case of Africa, have diverse soil types, compositions, and properties. So standardized protocols need to accommodate this diversity while ensuring consistency in data collection methods. So this is where it places a challenge on the sampling methodology, that the sampling methodology must be able to capture effectively the variability we have in the continent. And for the one employed in South Africa, I think to a great extent, we can say it fairly captures the variability. Then we have variability in survey techniques. Historically, different regions have used varying different techniques and methodology. Hence, there is a need to harmonize these approaches, and that's why we have developed this methodology as is being used now at a continental level. Of course, training is needed, and local expertise, development of local expertise is also a very relevant factor to consider. Then data integration and complexity. Integrating data collected from diverse sources and methodology of course, could actually cause a lot of problem in when you want to look at the quality of the data. So inconsistent data quality or format can hinder seamless integration. So that's why we are trying to have a robust data management system that will be, and with the FDMT to some degree helping in resolving this, so that can help handle this kind of a complex data. Then we have logistic challenges. So conducting continental-wide survey requires significant logistical coordination, including mobilizing personnel, ensuring consistent sampling protocols, and managing resources effectively. So these are also very important considerations in terms of infrastructure, poor infrastructure like poor routes, poor networks, and even security concerns. All these things pose challenges. And lastly, of course, you know that resources are needed. Money is required to fully do all this. Then, what are the opportunities? The opportunities, and there are many opportunities in doing this kind of a continent-wide protocol for soil sampling. One of the is that we have improved data consistency, enhanced better data integration, enhanced comparability, so that the standard protocol facilitates the integration of soil data from different sources, and this integrated approach provides a more comprehensive understanding of soil characteristics and processes. This also helps increase scientific rigor and support for policy development. High quality integrated soil data generated through standardized protocols can inform policy development and land management decisions at the continental level, thereby addressing the issues such as soil conservation, agricultural productivity, and environmental sustainability. Another key component is capacity building. Implementing a continent-wide protocol involves training and capacity building efforts, which can empower local communities, researchers, and institutions to participate in soil monitoring and research initiatives, ultimately contributing to sustainable land management practices. So the exchange of expertise can lead to improvement in soil survey techniques and data interpretation. And lastly here, we have long-term monitoring programs. With the development of the SDMT and this baseline data across continental Africa, it helps to monitor, it helps to further develop soil monitoring programs to track changes in soil properties and health over time. This longitudinal data can provide valuable insight into the impact of land use changes, climate variability, and management practices on soil quality and productivity. So, conclusions and lessons learned. Standardized protocols helps to facilitate data integration, that is the fair data principle. So it means that in doing this, we must ensure that the messages are clear, simple, and straightforward. Use of different media in terms of video, digital tools, and Pan-African protocol. It should be attuned to local conditions, of course, and it is monitored through an online system. The amount of time and money that is needed to do this kind of survey is grossly underestimated most of the time. Training and capacity needs, as I've highlighted earlier, are also often underrated. The way we do things has changed. Now we do paperless survey where everything is digital. So we tend to find that older generation have difficulty in using the technology, but the youngs are more attuned with the use of such technology. But however, both still needs to be properly trained. Security and logistical challenge remains a problem. Poor road networks are widespread in Africa. Security issues are also many. And we also found out that not all unsecure areas are danger zones. And that is why it is important to work with local partners in all the different areas. In conclusion, finally, implementing a continent-wide protocol for field surveys can significantly improve soil data quality and integration, leading to more informed decision-making and better management of soil resources across Africa. First, I would like to appreciate ISRIC for organizing this, for leading and coordinating this committee of practice of African soil information users and producers, and for organizing this showcase event. Thank you very much. I also want to thank IITA, at this time my employer, for the opportunity given to me to do this kind of activity. Thank you very much.
Thank you, Dr. Samuel, for the interesting presentation. Thank you very much. Thanks to all our speakers for the insightful presentation today. And we may move to the questions. Can we work on the on the Spectra library development together? So Dr. Nandamiso, would you like to answer this question, please?
Yes, thank you, Dr. Crow. I think it's not a problem. He's more than welcome to work with us.
We have a second question for Dr. Albert. Can you clarify if the 1600 is liters or milliliters?
Milliliters.
Okay. And the third question is saying why AS amongst other contaminations? Can you
that's a very good question. You know, before I started my concentration on arsenic, I first visited the site and I took samples. In fact, it was something random that I did. When I did analysis, all of the other metals, or so to speak, potentially toxic element, were not much of a concern, but arsenic alone was 1,803 milligram per kilogram. So that was more or less like a disaster. We're talking about 1.8 grams per kilogram of arsenic. And even though the threshold is, the average one is like 6.83 milligram per kilogram. So that made me to concentrate on arsenic from there.
There's another question, Dr. Albert, for you. It says why the SOPs used in your, is there any SOPs used in your laboratory for the analysis of metals and related parameters such as pH, EH, EO, and etc., especially with samples with the humid wetlands?
So pH, we do the pH using calcium chloride. It's just a matter of, I mean, after the filtration in the glow box, you do everything in the glow box because you don't want air to get into it to interfere with the results. So after that, we just measure it. We measure by going through the calcium chloride method and using the pH meter. Then we also measured the arsenic. Arsenic we measure with the ICP. Measure with the ICP. And what was the another parameter?
It says the pH or the EA, the
EH.
Yeah, EH, EO also. Yeah.
The EH is measured using a probe. You use the EH probe. So you connect the pH. So the probes were three. One is connected to measure temperature, another is connected to measure pH, and another is connected to measure EH. So those specific probes were made to measure this particular parameter. Sorry for the first answer when I was talking about the pH. The pH, now the pH is measured automatically via the use of the pH probes together with the probe for temperature and the probe for EH.
Thank you very much for the answer. And we have another question, Dr. Albert. It says, how did you calculate the redox potential? You recommended increasing the mobility of the AS by reducing the soil pH. If we do that, where would the free ions go? Wouldn't they further pollute the environment?
Okay, so now we should get something clear here. How did we calculate the EH? The EH, redox potential, is measured automatically using the EH probe, like I've already mentioned. So it's measured automatically. So I don't need to go and measure it because the microcosm experiment is done in such a way that the EH is measured automatically. Now, when I was recommending that we need to keep the site at a very dry condition, this is the answer. The reason here is that when you subject the site to flooding conditions, now when you subject the site to flooding conditions, don't forget that under flooding conditions, where we are going to have more introduction of hydrogen. So in this case, proton is going to be consumed during hydrolysis. So when you have hydrolysis, you realize that under oxidizing reducing conditions or during flooding, we have more of hydrolysis and we have more production of hydroxyl ions at the expense of H. Now, these hydroxyl ions, which is negative, arsenic is also a negative ion or a negative toxic element. So this facilitates the free movement of the arsenic and it becomes more available. Now, another point here is that the arsenic that is bound on the surfaces of the arsenic had a high correlation with iron. In fact, the main concern here was that the arsenic was bounded to the iron because of the arsenopyrite. Now, because the arsenic is bounded to the iron, that means that whatever happens to iron will also happen to the arsenic. So when under reducing condition, we have more of iron 3 becoming iron 2. Now, when it becomes that way, the sorption capacity of the ion reduces. Thus, any arsenic bound on the surfaces will also become available for pollution. And other factors like the dissolved organic carbon, other factors like the silver, like the sulfur, and those things that I mentioned in that particular graph. Yeah, all these come in a way to affect the mobility of the arsenic in wetlands, especially.
We do have a question to Dr. Samuel. I would like to know more about the SDMT. Any link to that, please?
Yeah, thank you very much for the interest in the SDMT. At the moment, the SDMT is not an open source material, though we are trying to see how we can explore collaboration with other with funders to see if we can get more fund to further develop this SDMT into an open source app that can be available. But at the moment, is only used within the confines of the project.
Yeah, thank you very much. Professor Aziz, you may unmute yourself and please ask your question. Thank you.
Thank you, Dr. Pro. My question actually is for Albert. I was the person asked about the arsenic. Because it is known that there are several other metals that are actually associated with the mining of lead. You have the likes of arsenic, I mean, the mercury, the cyanide, the cadmium, and the lead. But good enough, we have established why they worked on arsenic. But the other question is it soil borne? Is it actually part of the contaminants in the soil or in the minerals where they have extracted the gold, or it is part of the chemicals used in solubilizing the gold itself? That is the first part. The second part, some of the things he said, as far as the issue of the redox chemistry is concerned, is actually a well-known fact that metals will be more available at a very low redox potential, and they become to be unavailable when the soil is actually not in redox potential. But the issue is, I would have loved a situation where they do a follow-up experiment on identifying potential plants that could be used to mop up those pollutants in the waterlogged environment. Because it's not in all cases that we have the option of really draining the soil and making it aerobic before we are sure the pollutants are actually not in available form. That good enough, it is part of the PhD. Maybe you should actually take a further step to ensure that they identify plants that could be used to mop up the arsenic from the soil.
Can you hear me?
Yes, we do. Yes, doctor.
That is a very brilliant questions. Those are two questions that I would like to address. So I will start from the first one. I think the professor wanted to know about more or less where the arsenic is found. And so now, where the arsenic is found, now with that question, we will answer it this way. The arsenic is actually bounded to the mineral. So the problem actually at the site is more geogenic, because the arsenic is bonded to the parent material, which is the arsenopyrite. Now, even with the fact that the arsenic, so the major species in the area are scorodite and arsenopyrite. Now, scorodite is a secondary form of arsenopyrite, which is formed via oxidation of the arsenopyrite, oxidation of the arsenopyrite. Then we get the scorodite. Now, so the major source of pollution in the area is that it is bounded to the gold mineral. That is number one. The second part is that apart from the species, when you do geochemical fractionation of the arsenic to see which parts of the arsenic or which of the binding forms of arsenic are available, is the arsenic bounded to organic matter? Is it bounded to crystalline iron oxide? Is it bounded to amorphous iron oxide? Is it exchangeable, or is water soluble, or is residual? Now, because the problem there is more of geogenic, we find higher percentage of the arsenic be bounded to the fifth fraction, the sulfide, which means that those parts are residual and they are unavailable for pollution. Then the secondly, the arsenic is also bounded to amorphous iron oxide. Now, because of amorphous iron oxide, so we have greater percentage being bounded to amorphous iron oxide, because that particular iron oxide forms inner sphere and outer sphere complexes with the arsenic. So some get into the inner sphere, which is locked, and some also get into the outer sphere. So it makes it more bounded. So two, the residual fraction and the amorphous iron oxide fraction. Then the second question about the plant. Now, I have tried, and I still try various strategies to reduce mobilization of the arsenic. And even so, if the arsenic is going to become, even if the binding, which is the arsenic, which is the iron, even if more arsenic is bound to the iron, I've tried different methods to reduce the availability of the arsenic. We talk about biochar. I've tried with biochar. I've tried with iron oxide. I've tried with compost. I've tried with manure, and I've tried with inorganic fertilizer. Now we realized that as for biochar, the answer is not straightforward, because under some circumstances it facilitates the release of the arsenic, especially when it is in wet conditions. Under some circumstances, it also facilitates its absorption. But in terms of the iron oxide, which I tried, you realize that more of the arsenic is bounded to the iron oxide, which is not surprising, because the species of itself is that if we have more of the arsenic being bounded to the crystalline, amorphous iron oxide fraction. So when you do the experiment, you realize that more of the arsenic will be sorbed by the iron oxide that we were using. Then about the plant, I have tried with native plants, which are sort of available plant going around the mining sites, which are Promolena odrata, Lantana camara, fern. Fern, for instance, is found to be able to absorb greater percentage of the arsenic, especially more than 1,000 milligrams per kilogram. So fern can work. And I've also tried with ryegrass. Ryegrass is also able to extract more of the arsenic from the contaminated field, especially if the place is more reduced conditions. So the ryegrass is able to sort of bind most of the arsenic into the root. So we find more of the arsenic, like 8,000 milligrams per kilogram, being absorbed into the root and not getting into the shoot. So we can make use of ryegrass to sort of reduce the availability of the arsenic to become available for food chain contamination. We can use ryegrass, we can use the fern plant, and we can also use the Promolena odrata, which is native around the area. I don't know if this answered the professor's question.
Thank you very much, Doctor, for the answer, the sufficient answer, I would say. I would say I think we lost the professor for a while, but then it's just for you to know that this is recorded, so you can watch it later and you can see the answer. And also you are both members of the CoP, so you can communicate further if you have more questions or if you need more clarification or collaboration. There is another question for Dr. Samuel from Elizabeth, Dr. Elizabeth. Are you using a management system tools to store and manage your soil data?
Yes, we do. Apart from the SDM team being a form of more or less a data management system on its own, we also have a more extensive data management system tool that has been developed by ISRIC and is being managed at ISRIC in the Netherlands.
Oh, thank you very much.
Okay.
There is a question by Dr. Patrick. Oh, this is again for Dr. Albert. It says, what happens to the AS once absorbed by ryegrass? What is the disposal method?
This is a very tough question, as a matter of fact. Yeah. It sounds simple, but many people sometimes ask me questions on this, and it's a very tough one, but I'll do my best. As a matter of fact, I haven't tried to see what happens to the ryegrass after it is being taken up after harvesting, because the ryegrass is a C4 plant and it sequesters a lot of the arsenic on the ground and prevent it from being taken up. So for me, as a soil environmental person, and also as a soil scientist and as a remediation enthusiast, we are more interested in preventing food chain contamination. We are more interested in ensuring environmental safety. So what method will prevent the arsenic from becoming mobile to pollute the environment? What method will sequester arsenic in the environment, also on the ground, and prevent it from being taken by the food chain? So, but then there are other methods that can be used. I haven't tried any of these methods before, I must be honest, but there are other methods. We can use incineration, where we sort of create a hole underground and burn the ryegrass, which are contaminated, especially the roots. But then there are also other criticisms associated with that, because people are talking about groundwater contamination. So we can use that incineration as a strategy to be able to solve the problem, and we can also dispose it by using it to sort of for maybe fuel, if it is possible. If it is possible, we can use it for fuel. Yeah, and but then these questions, this answer that I've given here, all of them are subject to criticisms, because I must say this is a very tough question, which I haven't really done an experiment on them, but it's something that worth considering in the future.
Thank you very much. We don't have more questions in the chat. I may ask once more our guest if anybody want to do a follow-up question, or if you want to have to, yeah, Dr. Professor Aziz.
Yes, it's just to actually give a contribution to what Dr. Mensah said, as far as the issue of what happens to the ryegrass. If I'm not mistaken, I thought he said the grass was able to actually assimilate the arsenic in the root. If it's in the root, then we are safe. It means it has actually accumulated all the metals from the soil and keep in the roots. And for many of these heavy metals, the most important thing is for us to really enjoy our food chain, we need to look at the chemistry of this soil. Most often than not, when the soil is aerobic and you are able to control your soil pH, many of these metals, they are safe. If the soil is not deliberately been the amount is deliberately not been increased by our anthropogenic influences. And if we do, ensure that you keep the soil pH managed. If your soil pH is around 7, you have many of these metals, they will be in insoluble form. And if you have a phytoremediating plant that can concentrate the metals in the roots, then we are very sure it doesn't get to the food chain. Just like he said, the fate of plants that assimilate the metals in their shoot system is usually an issue of concern on what is the fate of those plants. Do you use the same plant as a material to be plowed back into the soil? If you do, then ensure that your soil pH is well managed. If your soil pH is not too acidic, then we are safe to some extent. So manage the soil pH and ensure that you keep your soil aerobic. Then many of these metals, we are safe from it. And if you have a plant that can accumulate them in the roots, it makes it safer for animals. Then those metals will not enter the food chain. That is just my contributions to the answers I gave. Thank you.
Thank you very much, Professor Aziz, for the invaluable addition. Dr. Albert, would you like to comment further?
Yeah. I think the professor has summarized it. In fact, I will add on to what he said about the pH. pH is a very important parameter that controls mobility of all these metals. In fact, and metalloids. In fact, I've done an experiment where I used what is called the acid buffering capacity. The acid buffering capacity to study how pH regulates the arsenic into groundwater. And it is interesting to find that at low pH, the arsenic mobility reduced, but the arsenic mobility became high as the pH increases. It is so because as the arsenic is a negative ion, so as pH increases, you have more of hydroxyl ions being produced, and it facilitates the mobility of the arsenic. So when you're able to keep the pH managed, especially at low pH, for instance, and not because low pH also brings the problem with metal toxicity, but then keep the pH managed. And if you're able to do that, I think most of the toxic elements will be under control. Then also, in fact, as a matter of fact, it is so difficult to rely on phytoextraction as a control. In phytoremediation, let us most often concentrate on those plants that are capable of sequestering the heavy metals in the root without it being taken into the edible part. A typical example is maize. So there's something called substitution planting. In substitution planting, you plant that plant or that crop that can do well in the contaminated environment without its food chain being contaminated. So maize will also sequester most of the arsenic and the toxic element on the root, and it will prevent it from being taken into the edible part. So this, among many other ones we can't discuss here, I think will be able to help to curtail the problem with pollution associated with heavy metals in the environment.
Thank you very much, Doctor, for the addition. The final question in the chat was for all the speakers for the possibility of sharing the presentation. I'm not sure if that's possible because as I know that it's part of a big project for Dr. Samuel and Dr. Nandomiso, and for Dr. Albert, I don't know if your work is already published, so maybe if it's published, then you put a link or provide us with a link to your publication so we can share
They are
later.
published.
But yeah, I don't know, maybe Dr. Samuel and Dr. Nandomiso, is it possible to share this information? Is it available on Soil for Africa website? We can share the website link.
Sam, do you want to go ahead?
Yes, please. Yes.
Nandomiso, please go ahead.
Yes, please.
I think at this stage, like Sam mentioned, most of these management databases that we are still using are still under development. And some of the data we are still putting it together, so we have not polished it to share with the public as yet, but that's our goal. So some of the outputs, I think there are some of them in the website already. So if they can visit the website, they will see some of them. And hopefully by the end of 2025, or around May, when we are done, everything will be there.
Thank you very much. Dr. Sam, would you like to add?
Yes, thank you very much, Kru and Misu. In addition, if you also Google Search for Africa, or go to YouTube and Google Search for Africa, all the protocol is available on YouTube, and you can freely download. Major part of it are in form of videos. So, and in different the good thing is that it's in different languages. You have it in English, you have it in French, and you have it in Arabic. So you can freely sit there. Also available on the website, particularly when you go to Search for Africa page, just click on the Fit Campaign, and under the Fit Campaign, you see all these materials from which we have extracted some of this presentation from. And they're also in English, in French, and in Arabic.
Thank you very much. So for all our guests, we will share the link for Soil for Africa website and the link to Dr. Albert's publication later when we publish the video. So you can find everything in the description also for the video on YouTube. I will also share it on the WhatsApp group for the COP members. And I would like also to invite our guests, if you are from Africa and you're still not a member of our COP, please visit our website, isric.org. On the community of practice section, you can fill in the form and join us. Thank you very much. On behalf of ISRIC and COP teams, I would like to express our grateful for all our speakers to join us in today's event. Thank you for your time, energy, expertise, and sharing your latest work in this event today. And for our respectful guests, thank you very much for joining us today, and we hope to see you again in the future in another showcase for the soil information system for Africa in different domains next month. Thank you very much again, and wish you all a great day.